Ok. Y bueno, un par de cosas antes. Está bien si sienten que su ancho de banda se ve perjudicado, pueden apagar su cámara, pero si tenemos más cámaras prendidas, creo que es mejor, porque entonces así Pamela no ve un montón de cuadros negros, ¿no? Y hay un poquito más de interacción, como si estuviéramos en persona. Y lo único es si empieza, o sea, quizás los micrófonos sí hay que apagarlos, pero si tienen preguntas, si quieren interrumpir, encienden el micrófono y pueden interrumpir. Y bueno, Pamela es un postdoc que lleva casi un año trabajando en el grupo de Aurora, y antes de esto estuvo en la UNAM en México, ¿no?
Sí. Sí, por ahí habla un poco de eso. Pero sí, sí.
Y bueno, nos va a venir a hablar de los proyectos nuevos que está haciendo en el grupo de Aurora, de mediciones rheológicas en glóbulos rojos.
Okay. Bueno, gracias. En inglés, ¿no?
Ya.
Vale. ¿Ya pueden ver mi pantalla? Can you see my screen?
No.
No.
There you go.
No. Well, thank you. I will talk to you about the project we are developing at the Interfacial Dynamics, Nanotechnology, Fluidics, and Biophysics group led by Dr. Aurora Hernandez. And the title is Biorheology within Microchannels as a Diagnosis Method. Well, first of all, biorheology is the science, or refers to the science, of the formation and flow of biological systems or matters or materials related to biology. This project is a kind of a collaboration with UNAM and Universidad of Barcelona. Okay, so of course, if we talk about rheology of important biofluids, but the most important biofluid is, of course, blood. So it has kind of a special name even for the rheology of blood, and it's called hemorheology. So here I will present some interesting facts that I believe you can associate with the project directly, because we're trying to make a diagnosis method based on the rheological characterization of blood flow. And it has been done, this characterization, for a long time ago. So here I can show you, can you see my arrow?
Move it.
Do you see it clearly?
Yes, we can see
okay.
Your arrow.
Well, okay. Then we have the red blood cells that are like the semi-solid part of blood. So what they do is try to see what is the effect of modifying this shape of the blood cells, and this can be done very easily because they are permeable to water. So if you put the red blood cells in a solution that has a high concentration of salt, they will dehydrate, right, and become like these shrink things. So you can see here that viscosity as a function of the shear rate. So this means that if you put a force that is shearing the blood and kind of increase this shear force, and you measure the viscosity of this sample of, let's say, normal blood, we see a clear shear thinning of the viscosity of the blood. This means that it decreases as I go to a higher shear rates. But if I put these red blood cells and increase the amount of salt in the surrounding solution, then they will start to shrink, and then this shear thinning will be not so pronounced. So this means the cell would be not as flexible as it is in this case, but because it's only starting to be like a fat, empty bag, right? So this clearly tells us that this characterization can tell things about how our red blood cells are behaving, or the state in which they are. Another interesting thing that has been reported is that not only the red blood cells can be tested or characterized with this shear stress or rheology measurement. Okay, here we see the viscosity of blood of two individuals. These are two different healthy individuals, but we see as these shear bands, and each shear band has an associated component of the blood plasma that can be, let's say, affected. Let's say if you have some alteration of the plasma proteins, maybe this shear band will be most affected. But if you go to higher shear rates, then you will see the effect of only the blood cells, because everything else is just like neglected. And the higher weight, let's say, for the viscosity is only related to the red blood cells. And this makes us think that if we have a big shear rate range, we might be able to see changes in the rheology related to these things, inflammatory factors, cholesterol, triglycerides, that are related with the health of an individual. So we think this is a very promising method of diagnosis. But these experiments were done in what it is called shear stress control rheometer. And I present you to you here. This is a really big machine. It is very precise. It is a really high-tech instrument. Of course, it has some disadvantages. That is, it requires a big installation. It requires a compressors. It is a bit expensive. And the sample volume that it requires to make the measurements can be of milliliters. And if we are talking about the diagnosis in the clinical environment, well, there are some samples that cannot be so large. Let's think about babies or people that can have hemophilia and cannot be extracted so much blood, right? Of course, these are widely used and they are well characterized. They have a high sensitivity, and they can be used to measure a wide range of materials, even human skin. So, but the most, the interesting thing, or one of the details that are not so well known maybe, is that water is very, very difficult to measure in these instruments, because as the viscosity is so small, they are not so sensitive to this. So blood, it's not a very high viscosity fluid. So this is why maybe there's an option that can be easily implemented in the clinical trials or in the hospital's environment. This is a microfluidic chip that can be, let's say, the alternative to this instrument. And especially, well, in the lab, has been a while that they developed this kind of instrument to measure viscosity in a microfluidic device. Well, I will show you just a little explanation of this. But basically what we have here is a microchannel where we can have a field of fluid and have the liquid and air interface. We can make this tracking of the interface movement as a function of time, but it will be dependent of the pressure gradient that we impose, and this is controlled by the height of this fluid column, and the height, let's say, can be associated with a pressure. Of course, we have here a tube that is plugged in this container of filled water, let's say, or blood, and it is plugged into the microchannel. So if we have the resistance in serious consideration into the pressure gradient, we can kind of make this association, of course, thinking of the movement of the fluid in one direction with no components of the velocity to the walls, only in the flow direction, and working in this stationary velocity profile approximation. So let's see this equation that is trying to express the relationship between the stress, the shear stress that is related with the pressure gradient and the geometrical factors, and the shear rate, that is the velocity at the center, the maximal velocity, divided by this height of the channel. And of course, this is a power law. If we see this relationship plotted here in this graphic, we can see that, well, with changing the height, we can have different effective pressures, and for each pressure that we put, we can measure the velocity within the microchannel. And if we make the fit, this will give us the exponent n. And when n is equal to 1, this means that the fluid is Newtonian and the velocity is constant. But if we have n exponent less than 1, then the viscosity is has a component, a behavior non-Newtonian, and will be shear thinning, as we saw for these graphs. On the other side, if the exponent is greater than 1, it will be shear thickening. So just by using this equation and the pressure and velocity measurements, well, the shear thinning behavior of the blood of different days and of different percentages of red blood cells diluted in plasma can be characterized, as we see here. And of course, plasma, it's got no elastic or component, as the red blood cells are missing, and it has a constant viscosity. So following this idea of the red blood characterization and its dependence of the content of red blood cells or even the days, we kind of fuse this project with some things that I was doing in Mexico, as Antonio said. And I would like to make a brief parenthesis and talk about how we got to this mixture of projects or collaboration, because I don't think I've ever talked about this. And Antonio said it was a kind of a free speech, so I will take advantage of that. And let's say that I'm a biomedical engineer, and I really spent a lot of time working on physiology, working with rats, and we made a kind of approachment that it's kind of common, and we saw the cardiovascular system of a rat like a black box, and we just put a periodic inputs of a drug to control the blood pressure, and the output to this system, the rat, was blood pressure. We made a model, and well, that worked pretty well. But we started to think about microfluidics. It was kind of a rising technology. So, and we didn't want to kill any more rats. So what we thought was this system, and I think it started all, is like an artery on a chip. This is the artery. And of course, the artery has blood pulsating within it. So the idea was have the blood vessel here tied to the microchannel, put blood, and oscillate it. But before doing that, we had to go and be a bit more into microfluidics, study some fluids pulsating as Newtonian fluids. That was the easy. Then before going to the blood, we had to have a model of a viscoelastic fluid, because blood, as I said, is a viscoelastic fluid because of the presence of the red blood cells. And in all of that, well, we never put blood in the channel to oscillate, because, well, many interesting things arise, but the whole idea was pulsating blood. And then just by chance, I got a sample of blood from a rat and make dilutions and put this to oscillate the same as I did with the rat, but with a controlled oscillating input to the blood, and recorded what happened to the velocity. And well, this is not a result that can be just published or anything, because it was just one sample, but it was like the moment that we could fuse all the things that we already had done and try to see if something happened with blood and frequency. And well, at the end, I would like to highlight some of the things of this experiment that I hope you can see to match with the project that we are developing here. And we have a pressure measurement of the driving of this interface, the interface tracking. And of course, we have an additional component that is the frequency. So Dr. Aurora gave a seminar back in Mexico, and along with Dr. Eugenia Corbera of UNAM, we kind of talk about this, and this project like rise, and we entitled this Biorheology within Microchannels as a Diagnosis Method of Anemia. And what we have here is the same as it is already done in the lab, an interface of liquid oscillating. We track the interface, and we have a recording of the pressure that is driving this interface. And based on the measurements of velocity, position, pressure, we can make some estimations of the viscosity. And as we can now have access to blood, to human blood at least, we can make these dilutions and see what is the effect of putting 10% of red blood cells in plasma, 20%, 30%, and so on. So the experimental setup that we have now installed here at the Universidad of Barcelona is shown here. Well, this is the microchannel. We have a cylindrical microchannel that is plugged to this part. This I call the oscillation generator. This is a piezoelectric that moves or makes an oscillating push and suction. As you supply it with any electrical periodical sign you want, we put, of course, a sinusoidal, and it kind of it behaves like a motor, and this is like a piston. This goes plugged to a elastic membrane, and this goes to a cylinder and then to the microchannel. On the side part, we have this small tubing that goes into this, that is a pressure sensor. If you see here, I put blood, it's all the red fluid. So all the system is filled with blood. The diameter of the microchannel is 500 microns. And the diameter of this small tubing is 125 microns. So as in the case of Claudia, of the square microchannel, and the experiment that I just showed you, this resistance is going to dominate, and that will make that the velocity is not dependent of time. I will show you this in the next slide. I hope it's a little bit clear. Just to be a little bit more specific about the setup. So here in the drawing, in the scheme, we can see the oscillation generator, the piezo actuator, the elastic membrane, the system field of fluid, the pressure sensing, and well, here's something that we add. As the microchannel is a cylindrical or a cylinder, we have some cylindrical aberrations, and we erase that or fix that to record it with a high-speed camera by putting the sound that we want to observe in a small, we call it a small pool filled with sugar, with a sugar in water solution. So this matches the refractive index of the PTFE or Teflon that it is made of tube. Well, one of the things that I would like to highlight of this setup is that, well, this is a microscope, and it is a small plate. The whole setup, this is 15 centimeters, and these are 8 centimeters. It is all fixed, so it is portable. You can just take it out of the microscope and take it to another. The tubing is replaceable, so you can just pull it out and then put another one in, so it's cheap. The volume sample that we can put inside is 40 microliters, which is a really small amount. The channels are disposable, so if we put blood, we don't have to clean it, you just throw it away. And one of the things we think it's the most interesting is we can test several shear rates at once, because, well, we are going from 0 to a maximal velocity, or from a pressure of 0 to a maximal pressure, then going back. Well, in the next slide, I will show you a little bit more. And another possibility of this is that we might be able to work with something that is called in the micro rheometers as SAOS. It is small amplitude oscillatory shear, and it is what I show you here. I couldn't find a better video, but if you see this tiny movement in a oscillatory rheometer, it's just doing this frequency imposition to the sample, and here we have almost the same. Of course, we are not yet quite there yet, but it is a possibility. Okay, so here I show you the interface. This is water. The white part is water. This is air. Well, here are the walls of the cylinder. And with this dashed line, I show you the tracking that MATLAB does. in the image analysis. So if you see here in the video, well, the interface bends, then it goes to the other side, it flattens, and then goes back, and so on. So this is all the data that we have, the position in time. And if you see, we have the position, but we also can calculate or estimate the contact angle at each time. Okay, here I show you the position data in time. The black line are the tracking from the MATLAB. And as you see, there are a bit of square areas. That's because, well, there are sometimes the interface is not clear enough. So if we make a fit that is practically the same, we can work with this. Here I show you the pressure that is recorded by the pressure sensor as a function of time. We can also do the same. And here in red is the contact angle. Also, the one that is associated with each image. This contact angle we can use to estimate the capillary pressure as depicted here. And then we can, from the difference of the pressure of the driving, that is the piezoelectric pushing, this pink line, and the capillary pressure here, I show you this in the blue line, again obtained by the experimental data. Well, we obtain a total pressure, and following what it has already been said or established in the lab, we directly use the one-dimensional approximation. And well, let's say we estimated the velocity that we should obtain using this, let's say, Poiseuille law, including the terms of the resistance of the tubing in the sensor, that, as I said, was the dominant one, and then the resistance of the microchannel is neglected. We can obtain this expression. And well, if you see, here there are no other terms that depend on time. Only the pressure, the total pressure could give me one velocity. And let's say, for instance, 1 point. I will have one pressure and then the associated velocity. And instead of trying this pressure gradient and then clean the microchannel and change the pressure and put a higher pressure, make the measurement of the velocity, and then try another pressure at once, I will be able to do continuously. So each pressure could be the same as putting the flow in a constant direction. Well, this was what we were thinking, and when we compare our experimental data, let's say I take my position experimental data, I derive it, differentiated numerically to have an experimental velocity, and compare it what is estimated, and the results are very, very similar, as I showed you here. The pink line is the experimental velocity, and the dashed red line is the estimated velocity. But what we wanted to do is, well, estimate the viscosity. So for doing so, we have, well, our collaborator, Andreu Benabet, helped us and came out with a very good idea of what if we have this equal. So we have the experimental velocity, we have these measurements, and we only solve for the viscosity that makes them the same. And if we do that, I show you the data for 8 experiments. This is 1 millipascal second, the viscosity reported for the water. And these are the estimated viscosity by comparing of the both velocities. As you see, well, there's some noise, but it's pretty accurate, as we think. Well, even more, if I define a shear rate, as I have the velocity in the center of the microchannel, and I know the radius, well, I have the measurement of the pressure gradient and these geometrical factors. Well, I would expect to build this well-characterized sort of the most known graphics of viscosity as a function of shear, as I show you here. Of course, there are some issues with the smaller velocities, but the tendency is very, very clear. So we think we have a good viscosimeter, at least in our system, and it's working now for water. And then by making or being sure of these measurements, we can go and work with blood. Here I show you some of the recent experiments. This is plasma. This is 10% of red blood cells diluted in plasma, in its own plasma, of course, 20 and 30%. All of them are oscillating at 1 hertz. The videos have been slowed down, so this is not the real time. But you see, the interface is very clear. It is oscillating. Of course, here on the diluted sample, you can see some of the movement that is related to the red blood cells. Here's 20%. And 30%. Of course, working with blood, it's a little bit more difficult. There are things that we are now exploring. We have been doing these experiments for a little time now, but I think we are really into it. We are now working full time with this, and we might be able to show you what's going to happen because we are not there yet. We have tracked the position. So as you see, it's a very sinusoidal wave. This has no trouble with the tracking of the interface with the image analysis. The pressure sensor and the shape of the pressure is also very sinusoidal, as it was in the water experiments. So we are obtaining very, very good signals to analyze. Okay, and then the things we have to do from now on is to measure the viscosity in all the dilutions to make some, let's say, curves of reference. And then from these curves of reference, we can make some comparisons of samples that are of people, and we can classify them as, I don't know, 20% hematocrit or 25, and that would tell us that this person has anemia. Of course, we have not yet explored or fully explored the change with frequency, but this is a very important thing that we have to see now, because as I showed you in the first graph, there are some shear bands that could help us to determine or to differentiate between some changes in the concentration of blood, of sugar, of triglycerides, and maybe this could go even more and have applications to many other diseases. Of course, not only the shear rate band is flexible or allows us to do this testing. Also, there are other biofluids that can be tested for diagnosis. There are some reports that says that, for example, the maternal milk has some change in the viscosity if the mother is infected by Zika, a virus. So at these times, it is very, very important and interesting to see that. And the last one that we are very, very interested in is the oscillatory shear stress. This protocol is already oscillating, and we have to carefully see if we can associate this with what it is done at the macro scale, and maybe measure the complex modulus with this elastic and viscous component. And that would make the whole diagnostic method a complete rheometer, the one seen, well, in the labs. Well, so far, this is what we have done. If you have any questions, please let me know.
Thank you very much. Does anyone have questions? You can just unmute your microphone and go ahead. If not, I have a couple.
Okay.
Right, so something struck me as odd. The blood, so the water has a very big change in contact angle, but the blood doesn't. Is there any reason why it happens?
What is the last?
Do we know why?
We still don't know why, but it might be something to do with the wetting properties of plasma, you know. Of course, we have PTFA or Teflon as the material for the microtube, but we also have the same geometry and the same size in glass microchannels. We are still working on trying that to see if it is something to do with the wetting or something that I don't know. Maybe the proteins are it's called adsorb at the surface. So they are just like making a wall of proteins that is maybe changing this. Yes, you're right. You're very
It's weird, no? That even if it's moving at the same angle. It goes like so, like this in this direction. This is not intuitive, but
it changes a bit. But if you see
not
it's
as much as in water.
Exactly. Yeah, we don't know. Well, there are things that we did not expect, of course, but we have some troubles when changing from a micro to a micro scale. And one of those is the surface tension becomes very, very important.
And the other thing is that you put all the deviations from a sinusoidal as the aberrations as problems with the tracking, but a lot of the sinusoidals look like they just don't have a linear response. Could it be, for example, these features that you see in the sign, you have like a little bump and you say, oh, this is the tracking. But it could be that is just the actual response, because in other cases you see like things that go like a bit more pointy than a sign.
Yeah, well, the first step was trying to see if we could say the same as in the one direction experiment. Then changing the direction and the things associated with that is something that we have to be, careful and look like really go into the details. But as a first sight, it was really, we were very happy that the velocity calculated only with the pressure gradient matched the velocity of the experiments. So it makes us think that we are going in the right way, of course, with no details yet, but they can be added in the way.
Does anyone else have any questions? All right, so, well, yeah, one last one. Sorry.
Okay.
When you say artery in a chip, did you actually take an artery from a rat and put it in a chip, or is some membrane that acts as an artery?
No, I actually took an artery of a mouse and put it on a chip.
Okay.
Well, yeah. Yeah, because, well, it really is what it started off. Well, you try to go to the most alike system, but it is a living organism. Of course, in humans, you cannot do that. But, well,
but the point was to see the elastic properties of the artery or of the blood?
Yeah, the thing is was going to pulsate a viscoelastic fluid in a viscoelastic artery. But it was too much.
Yeah, it was
too
many things at once.
Yeah. So this is going in this way of understanding things and then going to the more complicated ones.
Can I ask, Pamela, where do you have better results at detecting the front or the edge of the fluid? With blood, plasma, or water? I mean, with this,
what do you mean with better?
Answer to detect the position for which fluid?
Well, we've been working with water as for the start. So of course we have been working on that for a longer time than on plasma. So just by time spent on that, it is water.
But in terms of transparency and actually detecting the position, because as you are doing it in optical methodology or optical methods, I mean, I think that blood should be easier.
Yeah, it should, but well, you have cells, and sometimes they get to the walls, and I don't know. Sometimes it has gave me problems with this blood sticking to the edges. But well, we're working on that. We haven't not worked so long with blood to say this is better or we cannot do it with this better to do this, So with blood, it's now starting everything.
So what you're saying is that it leaves like a residue in the walls, and that.
Yes, sometimes it does. Yeah. It's not sticky.
Okay, sorry, does this residue that you're talking about, does it affect the measurement you're taking, or its effects are negligible?
The what? I didn't hear you.
The residue. You're telling me that there is a residue as the fluid. If I understand correctly of what you've been saying, is that as the fluid moves, it leaves like residues on the wall as it oscillates. I mean, does it dampen somehow subsequent oscillations? Does it have an effect that you read on the instruments or something like that?
Well, I can make some things in the experiment to make the optics a little bit better, because at the end, when I processed the video, I only had to put a threshold of gray to distinguish between what is white and what is black. And sometimes even if there is a trace or some red blood cells or some of these stains on the walls, maybe if they are not opaque enough or dark enough, they can be just neglected, and the threshold would be enough to to make it transparent and put it all in white and the interface in black, and then have the interface done like this. But of course, we are still working on that. It is better to do it on the experiment than do it while you are processing the video or analyzing the video.
Okay, thank you.
Okay, so if there are, unless there are something more pressing by anyone, thank you very much for coming. One more thing is that I want to take the advantage of people who have not been in the seminars on other occasions. If you are interested in this kind of thing, please subscribe. We have a mailing list. We do this every 2 weeks, and also if you want to give a talk about general soft matter ideas, you're welcome to contribute. Thank you, Pamela, again.
Thank you.
Thank you.
All right. Bye.
Thank you, Pamela.
Thank you.
Bye.